everyone. Welcome to today's live broadcast, a new generation in immunoassays presented by Dr. David Bourdon. I am Marjorie Torres of LabRoots, and I will be your moderator for today's event. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this presentation is interactive. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we will make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right of the Q&A button in the lower left. I will now turn the presentation over to Dr. David Bourdon.
Hi, everyone. Thanks for joining today. I'm really excited to be speaking with you today. My name is David Bourdon, and I've been with the company for about 8 years, focusing on next-generation immunoassay platforms across the biomarker discovery as well as validation spaces. I'm very excited to speak with you today about an innovative platform that we've developed over the last several years. This work was conducted in close collaboration with multiple interdisciplinary groups across our organization. And also what's exciting is this is also the product of a lot of very close interactions with various immunoassay users, not only in the US, but across the world. And so through that interaction and through that sensing and asking people what they really needed and working with people in the translational research space or biomarker discovery space, my colleagues and I have learned a lot about what people need these days, what their demands are, and what's going to make their work easier as well as more powerful when it comes to publishing and basically furthering biomedical research. I have the pleasure of sharing this platform with you today, but let me say that this platform and the innovative work behind it is really the product of a lot of hard work from many people, not only the talented research and development team that I lead, but also my close colleagues across the gamut, product management, marketing, program management, as well as leadership. R&D leadership and business leadership have all been very supportive of this technology, and I really want to express my appreciation and just say that what we're talking about today is a contribution of a lot of people. So I want to say hats off to everyone for this great work. So let's get started. So in slide 2, what we see here is basically just a wireframe diagram on the left side of the immune system. The various cell types and the extracellular mediators, cytokines, growth factors, etc. My background is in GPCR signaling. So when I look at a wireframe diagram like this, I get excited about all the different ways in which these extracellular mediators can play. It's definitely not a single linear process, right? Many of these inflammatory modulators have multiple roles, and I think that's what makes the immunome and studying inflammation very exciting, but also very challenging. And so on the right-hand side in this slide, I'm showing a number of different immunoassay tools that our company has developed, and I've had the pleasure of working with different talented teams focusing on the Western workflow, bead-based immunoassays from the ProQuantum and other Luminex assay platforms, as well as flow and imaging. And so let me just say that my goal of showing this slide is really to impress upon everyone to use the right tool at the right time. We have a lot of different immunoassay tools out there, but they're not all ideal to be using all the time. And so I think even today's presentation is focused on when to use those right tools, and I'll get to that in just a minute with the singleplex. But a lot of the translational researchers and biomarker discovery enthusiasts have adopted this discovery to validation workflow in which one uses a high-content technology, for example, maybe mass spec or a high-content protein microarray to discover targets. But then as those targets get verified with other orthogonal platforms and other maybe functional assays, the relevance of that target and whether or not it's truly involved in the particular physiological process that's being studied is further refined. But then it sort of, I think, let me just say that the ultimate significance of a biomarker or a particular analyte that's being associated with a disease progression really gets fully vetted at the singleplex, or what I'm calling the validation step here. And that step is where basically the technology that's used is a singleplex technology that can be done in high throughput. It can be done very rapidly. It needs very little sample, so you're not consuming a lot of your precious sera, for example. And that's where, if you look at this discovery to validation diagram here, the singleplex platforms are where there's been a lot of activity around trying to make more innovative and more novel platforms. And I'm sure you've seen a lot of activity in the last couple of years in this space. There's been a lot of activity, but there's still a lot of needs have gone unmet. And so my colleagues and I have set out to really execute upon this to make sure that the singleplex platform that we deliver addresses a lot of those needs. And so today what I'd like to unveil is the ProQuantum Immunoassay Platform, the high sensitivity immunoassay platform. And what's wonderful about ProQuantum, and I really am excited about this because I think a large company like Thermo Fisher Scientific has so many areas of expertise. And but even in our space, it's very easy to kind of work in a I wouldn't say a silo, but very easy to work within a certain discipline. But really, I think some of the greatest innovations and some of the greatest contributions are when people step outside of their initial comfort zone and think of new ways to tackle challenging problems. And I believe that's what we've done here. So what ProQuantum does is it combines the power of matched antibody pairs with the sensitivity and broad dynamic range of qPCR. And it's important to note here that in our company, we're well known for our proteomic technologies and, of course, our TaqMan genomic technologies. And so naturally, putting these two key areas together, it's bound to address some core needs. And these needs are as follows in the latter part of this slide. So first of all, high sensitivity. For many of you out there that are looking for early detection of inflammatory mediators, you know that it's very important to understand very subtle changes. We're talking femtograms per mil changes between, let's say, a disease population and a healthy age-matched control. So what we've done is we've produced a platform that gives great sensitivity. I mentioned just a minute ago about sample consumption. We know that many of these samples are very precious. They're irreplaceable in some cases. And so being able to get a lot of great data, very powerful data out of a very small sample is a huge contribution. And it's something that everyone asks for because, of course, if you have a limited amount of sample, you want to get as much great data out of that sample as possible. And what we've done is we've addressed that with a sample volume of between 2 and 5 microliters. And I'll go through in just a minute as to why there's a range there. But again, as low as 2 microliters for multiple replicates for a readout. This third bullet point, fast and easy workflow. This is something that I think, I know it's semantics, but many times people talk about sample to result, and I see a difference between result and answer. The result may just be the numerical value, but the answer really is whether or not something is statistically different than another. And actually what I'm going to do today is I'm going to provide a live demo of our new innovative cloud platform so that we've designed our own software to go with this platform. And I'll show how it works in just a bit. but being able to not only set up the assay and run it, but then also get your final answer, get statistically relevant comparisons in a very short amount of time is really important for you to be able to understand what experiment you do next. Broad dynamic range, that is something that I think anybody who's run an assay that maybe only has 2, maybe 3 logs of dynamic range knows that if your unknown doesn't hit your standard curve the first time, what's the first thing you have to do? Rerun that sample at a different dilution. And what we've done is we've provided a platform that easily has 5 logs of dynamic range. So what that helps ensure is that the first time you run this assay is the last time you have to run it for that particular study. Just as important is with a lot of these new immunoassay platforms, many of them require a very expensive instrument. And what we've done is we've capitalized on the fact that nearly, I'd say, many labs have qPCR instruments, and this platform runs on real-time PCR instrumentation. So even if you don't have the instrument in your lab, chances are somebody in your hallway has that instrument, so you can actually try it for yourself and see how this platform works without having to request a demo or purchase an expensive instrument that can only be used for one type of workflow. And then finally, this cloud software. This is an area where I think myself, as a protein biochemist by training, it's really exciting for me to work with skilled software developers in our company who've helped take our vision of what we want the workflow to look like and what the end user needs and how quickly they need it. We've been able to make that reality, and I hope to show you that in this live demo, and I think you'll be very impressed. Now, first of all, let me just kind of go through at a very high level, what is ProQuantum? It's a proximity-based assay platform in which you still have 2 antibodies. One antibody is conjugated to a 5' or a 3' oligo. So there are 2 different epitope-specific antibodies, each with a different oligo conjugated to it. And so just like a traditional immunoassay platform, there are 2 epitope-specific antibodies as well as the analyte of interest. So what happens is, upon mixture of the analyte in your sample and the 2 epitope-specific antibodies, these antibodies basically bind analyte, and the oligos become stabilized. Then in the presence of DNA ligase and cycling conditions, basically the 60-mer and the 40-mer basically create a single-strand DNA template that can then be amplified through 40 cycles of qPCR. So essentially, the more analyte in the sample, the more bridging of oligos occurs. The more bridging of oligos creates more qPCR template, and the more qPCR template leads to a faster amplification of the template strand. And so this all uses TaqMan genomic technology, and I'm not going to go through all of it in the interest of time today, but essentially the more DNA template you have, the more fluorescence you produce in a shorter number of cycles. So I will be referring to CT, which is the cycle number to reach threshold. So again, the more, the more analyte that is present in your sample, the more antibody binding that occurs, and that's proportional, of course, to the amount of analyte. So you run standard curves just like any other immunoassay. But then the number of cycles to reach threshold is a lower number when there is more analyte present, and vice versa, as shown in this figure on the far right slide of, or a figure of slide 5. So what we see here is actually real data from our IL-10 assay. NPC is the no protein control. And so with no protein present, only the antibodies with the conjugated to DNA oligos, the system requires 31 cycles to reach threshold. But then as you titer in IL-10, the number of cycles to reach threshold goes down in a very predictable manner. Now, if we go ahead and subtract out the no protein control or subtract out the background, we get an inverse. And so basically this curve in the bottom right corner is very similar very recognizable to us in the immunoassay space as the beginning of a sigmoidal concentration response curve. And so it's a very predictable and very good fitting curve that uses 5-parameter logistic regression. And so the ProQuantum immunoassay kit, actually, there are a number of different benefits that we have worked on. The power of the assay platform I'll go through in just a minute, but also, like I mentioned, the simplicity. So we wanted to make sure that there are very few number of vials in the entire kit. And so what you see here on the left side is an actual photograph of the packaging, and then on the right side is a cartoon that explains what they are. So like any other immunoassay platform, there is a protein standard that is used to generate a standard curve, and then an assay dilution buffer that is used to not only reconstitute and dilute the protein standard, but also to dilute your unknown samples. Now, unique to ProQuantum, what we've done is we've kept antibody A and antibody B with the different oligos separate just to ensure quality. So actually the user combines antibody A and antibody B and the dilution buffer all together right before they use it. So that's just a simple, quick pipetting step to combine all three. And then finally, also unique to ProQuantum is this master mix and ligase. We've actually combined all the key components for TaqMan amplification as well as the DNA ligase, which helps create the initial first strand. So here's the workflow. And so at the top, what I'm showing is a typical ELISA workflow that most of you should be familiar with. You can see here the time frame, the number of steps, and I think what I want to point out here is what we've done is we've greatly improved upon the handling, the number of hands-on steps, and it's important to note right here that this is actually a homogeneous assay. So the first step in the assay is actually just to create your standard curve, dilute your unknowns, and place them in the plate. In this particular assay, I'm showing the 5 microliter reaction, which is basically, and I'll explain very briefly right here, the user takes their unknowns, 5 microliters each, and then dilutes them to 10%, so basically 5 plus 45. They dilute their unknowns and place them in the plate. Of course, also the standard curve is generated and placed in this plate. Everything incubates for 1 hour at room temp. Then with a couple minutes left before, the user takes the master mix and ligase, mixes it together, and adds it right on top of the existing PCR plate. There's no washing, no transfers, etc. So after the master mix is added, and it's a 40 microliter addition, after that, basically they reseal the plate, put it back into the instrument for the ligation and 40 cycles of qPCR. So at the end of the 40 cycles of qPCR, basically the CT values are calculated. So it's important to note here that this platform works with not only our applied biosystem family of instruments, but it works with any qPCR instrument. Any instrument you have that generates a CT value will work with ProQuantum. So after the 40 cycles of qPCR, the CT values are transferred to your cloud account. And by the way, if you have one of our new cloud-enabled QS3 or QS5 instruments, that information actually goes directly from the instrument directly to your cloud account, seamlessly. And so at the end of that transfer, it takes about 5 minutes, and actually I'll show you during the live demo how quickly the data analysis can occur. Now it's important to note here that this platform is also very flexible, and what I mean by that, I talked a little bit about volumes of sample. Now in this initial example here, I'm talking about a 5 microliter sample, but actually our kit is very flexible. And what I mean by that is if somebody doesn't have 5 microliters of sample, they can actually use a 2 microliter sample. So again, you would create your 10% sample, so you would take 2 microliters and you would add it to 18 microliters. And then 2 microliters of that 10% solution is used per well. So when you perform that type of workflow, our single catalog number will be enough material for you to run 2 96-well plates. So the way this works out, and the reason we did this, is we know some people really want to stretch their kit as far as they can. And so for those people, we provided the workflow. And by the way, our entire assay platform was developed on the 2 microliter volume. But if you're an individual that's just getting used to the technology and actually wants to up the volume a little bit just for pipetting comfort, you can still run a 5 microliter assay. We provide enough material for that. It's just that you only get 1 plate's worth out of the kit. And by the way, for people that are automation savvy or really want to stretch the envelope of their kit, this same catalog number will also be enough material to run 1 384-well plate. So at that point, you can see that the price per data point really goes down. And then also for people that have said, well, what if I want to run more than 1 plate at once? Are there options? Yes, there are. And this is a great innovation. And by the way, it's important to note that we have worked very closely with the side of our business or the side of our corporation that is the TaqMan Genomic experts. And so what we've done is we've designed this so that you can actually run this step 1 and this step 2 on multiple plates at once. And so basically, instead of just running 1 plate, you could, let's say, run 10 plates. 10 plates for the 1-hour incubation. And then what you do is you basically add the master mix and ligase, but only do a batch ligation. So again, 25 degrees for 20 minutes, followed by a ligase inactivation for 2 minutes at 95 degrees. At that point, the 2 minutes at 95, what has happened is essentially the antibodies and the analyte are completely denatured. And what's elegant about this is that from this point onward, it's nothing more than a DNA readout. And of course, because DNA is very stable, these plates, let's say the user ran 10 plates, they can all be stored and then run at a later time in batch. For example, if you're a user that has one of our twister arms that is basically working in conjunction with a Quant Studio instrument, you can actually read multiple plates at once. And because it's only a DNA readout assay and you don't need to be concerned about the presence of antibodies or analyte anymore, this can actually be done at a later time with greater flexibility. Temperature maintenance is not an issue at that point. So again, this is a great flexible option for those that are trying to get higher throughput and basically those that are trying to compare ProQuantum with other technologies that may have throughput benefits. And so I think it's a great solution for those users as well. So what I'm showing here is basically in slide 10 is a snapshot of our quick reference card. And so what you'll see from this quick reference card is that really gone are the days of having to read, a 10 or 20 page manual to get the gist of how the assay works. And so I think from seeing the next 2 slides, you'll say, wow, this is really easy. So the first step is to reconstitute the protein standard. For stability reasons, we provide lyophilized protein. And so on the side of the vial will be the reconstitution directions. And so the user basically takes a certain amount of the assay dilution buffer and adds it to the vial, and then waits for 15 minutes. And during that 15-minute time period, the protein is refolding, rehydrating, etc. During those 15 minutes, the user can then go ahead and start making their working plate. What I mean by working plate is essentially what we're doing is we're encouraging people to think differently about how they set up their immunoassays. I think we want to get away from the days of digging through an ice bucket and looking for your various random vials and trying to figure out which vial goes with which step and things like that. So what we've done is we've designed a working plate. And I know it seems like an easy concept. A lot of people just don't do it. And so what we do with a working plate is we encourage people to be very organized about the assay setup. Not only does it lead to better organization, but also it maintains everything cold. So we actually refer people to a little aluminum cold block that you store in the refrigerator right before you start the assay. So placing this 96-well plate on a cold block allows you to work with confidence that your proteins are not degrading over time at room temperature. Because also we know that trying to set a 96-well plate on an ice bucket and then keep the ice chips from falling into it is kind of a complication that you shouldn't have to worry about. And then finally, this working plate also enables multi-channel pipetting, and you'll see why in just a second that it's so much easier if you just take a little time to set up this working plate. So with the working plate, what you do is in this example, I will go through if the user was going to run the entire 96-well plate at once. So the user basically takes 12 microliters of antibody A and adds it to their conical tube. 12 microliters of antibody B, and then 696 microliters of the dilution buffer. All of this goes into the tube, a gentle mix through pipetting, and then even distribution across the entire 8 wells on this working plate. Now, the second step is to go ahead and find all those unknown samples. You know, many times people have them on different plates or different tubes. What we want to do is help everyone get all that stuff gathered up and then diluted before you even start. So basically the user will take 5 microliters, and by the way, they can use 2 microliters as well. That's an option. But 5 microliters plus 45 or 2 plus 18. So basically either way is a 10% dilution of all of their unknowns, and they go in this part of the plate. So after this dilution step of the antibodies and the unknowns is done, they're probably ready to go ahead and perform their standard curve. And again, this protein standard that was reconstituted basically goes right in here in the first S1 concentration, so standard 1, and then the user performs a traditional serial dilution down the plate. And as you'll see in just a minute with our cloud software, we provide a lot of flexibility for the end user to basically decide where they want to start their curve, what's the dynamic range, they want to stretch it out, maybe do a 10-point curve, the fold dilutions, the number of replicates, all of that is managed very nicely by the cloud software. So once the working plate is made up, and you can see this, actually, I went through and talked about it, but I think in the amount of time I was talking, a user could have finished half of it already. So basically, let's go to the next slide. So once the user has the plate set up, the working plate, the rest is a piece of cake, and all they have to do is basically pipette 5 microliters of the antibodies to every well on the PCR plate, followed by 5 microliters of protein standard or 5 microliters of unknown samples. After that's done, the user basically seals the plate, mixes it, and incubates at room temperature for 1 hour. It's during this 1 hour that the 2 epitope-specific antibodies are binding, the oligos are coming into close proximity, and then with a couple minutes left in the 1 hour incubation, the user simply adds 30 microliters of ligase to 5 mils of their master mix, mixes gently in a conical tube, and then pipettes 40 microliters with a multi-channel pipetter across the entire working plate, or the entire PCR plate. Then the user simply pops that plate into the qPCR instrument for a pre-programmed ligation, inactivation, and then 40 cycles of qPCR. Now this is the data you've probably been waiting for. So what we're showing here is some of the performance comparisons between ProQuantum Immunoassay Platform in red and a typical ELISA in blue. And what I'm showing here for gamma interferon and IL-6 is comparing ProQuantum with a normal non-ultrasensitive ELISA. But then for IL-8 and IL-1 beta, I'm actually comparing ProQuantum to 2 of our ultrasensitive ELISAs. And what you can see here is regardless of what kind of ELISA, ProQuantum definitely exceeds in the area of dynamic range as well as sensitivity, which is shown on the right-hand side here. So what I'm showing is analytical sensitivity, limited detection, between ProQuantum, again in red, and ELISA in blue for a number of targets that we've already validated. Now, what you can see is the platform's very powerful, but I'd like to also describe or share a couple differentiators that I think are really important here. And so as an assay developer, I've always been concerned that, okay, we make or acquire antibodies, we label them for our particular needs, but how do we know that what we do to the antibody is not affecting the specificity or the reproducibility of the assay? So what we did was we've utilized Siteclick chemistry, and this is a technology that we've owned for a while as a company. And it was something that I felt was being sort of underutilized, and especially in our space where we're performing TaqMan amplification, it's ideally suited for ProQuantum. And so what Siteclick is, it's basically a conjugation chemistry. It's covalent, but it basically targets the glycosylation sites in the heavy chains of IgGs. And so because of the chemistry, there is no way to alter the variable domains, which of course confer the specificity on an antibody. And so with Siteclick chemistry, we're attaching somewhere between 3 and 4 biotins per IgG molecule on the average, that is. And so of course that biotin antibody is then used with stripped avidin oligos to create our antibody conjugates that are then delivered in our ProQuantum kit. Another reason why Siteclick excites me is because what I show here is a degree of labeling experiment where we're actually measuring the average number of biotins per antibody molecule. This is across 9 different experiments. And what you see is a non-specific or non-targeted labeling method can give a CV of around anywhere nearly 50%. But with the Siteclick chemistry, you see here it's a very tight, very consistent 9%. And so it makes sense biochemically why it's such a tight value, but then somebody might ask, well, wait, how does a tight degree of labeling then equate to a better assay? And I think that's nicely shown on the right-hand side here. So what we see here on the right side is basically 3 different assays for IL-10 that were performed with non-specific labeling techniques, which are in blue, and then 3 assays that were using used Siteclick chemistry that are in red. And what you can see here is with the non-specific, sometimes you might get an assay that performs like this. Sometimes you might get one that performs like this. But in the case of the Siteclick, you get one that performs a very consistent across the board. And so that consistency is also, I think it's something that the end user should be impressed with, because what we're doing is we're passing along that consistency and that reassurance that the labeling chemistry now and 10 years down the road is never going to be affecting those variable domains. Okay, the next thing I'd like to talk about really quickly is basically, these assays, we validate these assays in natural samples. So we perform the spike recovery, we do linearity dilution, parallelism, benchmarking to ELISA. We do all of those assays, all of those validation steps that you have come to expect in other immunoassay platforms, and ProQuantum is the same way. And so what I'm showing here in slide 14 is basically a parallelism curve where I'm showing the overlay of various human serum samples along the standard curve, showing great parallelism, and then also not only for human serum, but also for PBMC supernatants, CD3, CT28, as well as PMA and ionomycin. And then on the right-hand side, I'm actually showing linearity of dilution for those same samples across a range. And so, again, that linearity of dilution helps ensure that regardless of the dilution, you're going to get very accurate results. And this is the same type of data for a different target, IL-17F. But again, we perform these types of validation experiments for every single target that we're releasing. And then here's just a very brief view of some of the benchmarking data for our various targets. So basically comparing ProQuantum on the y-axis
and ELISAs on the x-axis, showing very strong concordance between those two for a number of targets. And we perform, again, this for every target that's being launched. Now, finally, what I'd like to do is spend just a couple minutes touching on the cloud software. And I think this is something where early on, I didn't think that this was going to be such a game changer, but as I got more involved and other colleagues of mine that work very closely with the cloud software development team, we realized what a huge opportunity this is to help improve the workflow. And it's various steps in the workflow that other softwares may crunch the data, but they don't really turn the workflow on its head in a good way to say, you know what, how can we make it so much easier and make it different for the end user? And I think we've done a very good job of that with this software. So what I'm showing here are just a couple screenshots. I'll go through more of it in just a minute. But for example, we have a standard curve wizard that basically goes through and asks the user which kit they're running, what type of, the number of dilutions, the number of, the dilution series, the full dilution, the number of replicates, the number of points in the dilution, whether it needs to, the working plate needs to be set up in horizontal or vertical format is automatically adjusted. The user also is able to populate their unknowns in a number of different ways. And by the way, what we're showing here are 3 different colors for 3 different groups. And in fact, our software, which I'll talk about actually right here, is we provide that group-wise comparison. And this is a robust MSTAT modified Mann-Whitney group-wise comparison, because we know in heterogeneous patient populations, which is very much like what people are studying in the translational research space, they aren't a clear separation between, let's say, a disease group and a treatment group, et cetera. So a simple t-test or ANOVA does not suffice. So this modified Mann-Whitney group-wise comparison is very powerful. So let me not forget to go back up here and say that, a lot of people, when they decide what they're going to do in their experiment, they basically, will copy the protocol and scribble in it, or take notes and then, write things down. Everyone's got their own little shortcuts to try to document what they're doing. What we've done with this cloud-specific software is we actually go through and produce a customized lab instruction based upon how the user filled out this initial page here. And so that customized lab instruction can then be saved as a PDF, put in an electronic lab notebook, a physical notebook, etc. So our software, of course, provides all of the standard calculations for concentrations. Because this is a TaqMan-based readout, of course, we have all the amplification curves. We provide the 5PL curve fitting. For those who want 4PL, we provide that as well. We provide outlier detection for unknowns and standards based on certain criteria. And so let me just reiterate here that all of this is possible on any qPCR instrument. Even if your instrument does not create an EDS file, which is the case for any applied biosystem instrument, all you need is a Excel file with all the CT values in it. We can import that, digest it, and analyze it just like if it was a applied biosystems instrument. And then finally, let me just say that what we're doing is we're also making this software GraphPad Prism friendly. What that means is that you can actually save the data file so that it actually can then be opened up in GraphPad Prism, and then you can go ahead and make your various figures and graphs that you may need for publication purposes or a poster, etc. We know, myself included, I love GraphPad Prism, and so it's one of those things that making it easier to basically open up the file so you don't have to retype or sort of do an interesting copy and paste transformation to get the data in there. We've tried to make that very, very easy. So without any additional delay, let me go ahead and open up my browser window here so I can actually share my browser and show everyone how this really quickly works. Okay,
So let's get started. So what the user will see, so they've already got, let's say, they've already signed up for a free Thermo Fisher Cloud account. And so when they open the ProQuantum app, this is what they're going to see. Two choices: create an experiment or import view results. So if the user has already created a plate map, they would go directly here. But for a new user, everyone's going to get started right here. The first thing you see is, okay, a catalog number. So, for this example, let's type in our IL-6 catalog number. So A35573. Voila. You type it in, you get the species, the analyte. Of course, the lot number will be specific. The user can type that in. It gives a recommended stock concentration, the number of dilutions, the rate of dilution, the number of replicates, etc. Now, in this case, let's say a customer or a user or even somebody like one of our assay developers on my team, we really want to push the limits of this assay. So what do? We basically want to start the assay at a 5,000. We want to do a 10-fold dilution. So there you go. The working plate changes to a horizontal. And you know what? Let's go ahead and we'll still do triplicates. So now we move on. It says how the software actually goes through and says how many unknowns we now want. So basically you can go through, by the way, we always recommend a 10-fold dilution. The number of samples, you can actually choose singlet, duplicate, quadruplicate. And so basically if you wanted to do singlets, you can actually go ahead and type in all 60 here, and it auto-fills everything. But let's say, okay, the customer or the user now wants to basically, you know what, they always like to have a little bit of a gap between their standard curve and their unknown. So you can basically clear the selected wells, and it'll go ahead and you can adjust it accordingly. And let's go ahead and save that for the sake of this, let's just call this webinar. So what we're saving here is actually the plate map. Okay, so what I'm going to show here, this is the customized lab instructions that I spoke of. So basically, what we see here is is the quick reference card in a more customized format. So the first thing, chill the reagents. Whether you're doing the 5 microliter or the 2 microliter volume, the reason that's important is down here, whether you're doing a 5 microliter assay or if you're doing a 2 microliter assay, you see that the volumes, even for how to dilute your samples, it went from, for 2, it's 2 plus 18. For 5, it's 5 plus 45 right down here. The amount of sample required for the plate is already pre-calculated. Remember earlier, for a 5 microliter assay, it said 12 plus 12 plus 696. Well, we're doing just a little bit less than a whole plate. So you can see that these numbers have even changed. Now at the very bottom, you see the cycling conditions. And so, for example, a 15-second denaturation and a 1-minute annealing extension. That is actually for a standard block, or what we know as the 0.2 mil standard block. But of course, if you have the fast block, the 0.1 mil block, you can actually see even the cycling conditions go down to 3 seconds and 30 seconds. So again, these lab instructions are completely customized. You can save them as a PDF, you can print them out and paste them into your lab notebook, etc. Okay, so now the plate map has been saved, you've saved your lab instructions, you go off, run the experiment, and then basically you come back and upload the cloud data. And so basically you can import from your computer here, you select the EDS file, and here we go. We actually have the plate. We've already saved the plate map. We apply that plate map. So basically upload progress. The file was successfully uploaded. It actually gives, the software gives you one more opportunity because we all know we've been at the bench where we've done an experiment and then we decided at the last minute, you know what, I changed my mind. I want to do a different curve. I want to run different samples, etc. And by the way, so I didn't go back and renumber these, so it looks like this one's starting with unknown 13 and goes to 60. I should have renumbered them because I deleted this first row of singlets. But the user could go back and if they wanted to renumber those. But in the interest of time, I don't want to belabor this, so let's just go onward. We know that in this particular example, unknown 13 is our first unknown. So you click Next. Now, if I had changed something in the plate layout, it would actually the software would give me an opportunity to go ahead and save it under a different name. So maybe I call it Experiment 1 and then Experiment 1A if I decided. Okay, so here, it's already analyzed the software, or it already analyzed the EDS file. So in that time if I was talking, it basically took the plate map, overlaid it on the EDS file, and here we go. Here's the standard curve showing a wonderful dynamic range. At the lower and upper ends, we actually have red here. It's because the outlier parameters indicate possibly a CV that is greater than 15% or a recovery that's outside the 70 to 130. So the user can actually go through and decide whether or not they want to apply certain outlier parameters, or we have a more rigorous algorithm that was designed by one of our biostatisticians. But in this particular example, let's just leave it as is. So now we go ahead and just click Next and apply the existing. By the way, here's where we would also, if the user wanted 4 PL instead of 5 PL, you could also click on that and recalculate. So let's go ahead and click Next. And now what you'll see, okay, here's the curve still, but actually since we're not really interested in looking at that curve anymore, we've already validated the standard curve, let's go ahead and look at the data. So we can actually toggle this to look at all of the data table information. So we have theoretical concentration, measured concentration, percent recoveries. We can sort by basically the different standards, and what we're looking at here is the average view. You can actually select the replicates as well. And so basically what we're seeing here is S1 through S10. And so yeah, what you see here is S10 was actually 131%. So yeah, it was 1% over that 70 to 130 window. That's why it was flagged as an outlier. Okay, so you can go through here, scroll back and forth. You can actually look at particular unknowns. So let's pick an unknown here. And by the way, you can look at the AMP curves. So you can actually see the AMP curve for various unknowns here. And a lot of people that are very familiar with TaqMan genomic assays are very accustomed to looking at these AMP curves, and so they can be used for troubleshooting purposes or better understand subtle differences, etc. And so what's important to also note here is that if you want to download all the data into Excel, let's say you want to do something else with it, or you want the PNG files of all of the standard curves, so it'll actually go through and download all of those separate attachments for you. And so, yeah, I mean, this is pretty much it. You go through, and by the way, okay, in this particular example, all of the unknowns are in one group. But actually, you know what? The person might have said, you know what? I forgot to design my different group-wise comparison. So you know what? We can go back to plate layout, and let's go ahead and put this group, call it group 1. Call this one, let's say, group 2. And then call this one group 3. Okay, and so this is the classic example. Somebody runs the experiment and they say, oh shoot, I forgot. I should have listed the different group names. So now go back through, we can call it group. And what it's going to go back through is just basically go through that qualify standard again. But what I wanted to show is basically that group-wise comparison. So bear with me just for a second here as it recalculates. But what you can see here is in the amount of time that I've been able to import the EDS file, click on the various outlier criteria, etc., this is very rapid. And so what you see here now are the 3 various groups and the associated p-values with those groups. And again, this is the Mann-Whitney group-wise comparison. So again, you can see very quickly we get some very powerful results. Okay, so I'm going to pop out of this real quick, back to the slide deck. And what you can see here is I should be on slide 18. So again, I'd like to wrap up. I'm very excited. If you can't tell already, we've been able to share this with you. We first unveiled this platform just last month at the American Association for Immunologists meeting. We had a lot of great feedback from people at our booth and a workshop we gave. So again, I think this is a platform that meets a lot of needs. It's long overdue for people, and it's quick, easy, and very powerful. And so again, it allows that end user to measure very small sample amounts in a very small sample. And I think being able to get those answers in a very short amount of time is also just as important. And of course, that is made possible with a very powerful cloud-enabled platform. What you're going to see from us over this summer is basically, please stay tuned for a lot of early adopter stories as people start to use this platform. And definitely, come back and check us out. You'll see we're basically going to have teaser videos. This webinar will be made available in subsequent months. But definitely make sure to check us out at thermofisher.com/explore. And then also remember, don't forget to apply for your free Thermo Fisher Cloud account and start familiarizing yourself with the ProQuantum app. And definitely, we look forward to working with all of those end users. I'll be on the road talking about this. My colleagues have been trained on this, and they're very excited to share this with you as well. And so definitely stay tuned as you can learn more about this product in the coming months. So again, thank you very much for your attention. I hope you learned something from this and see ProQuantum as a next generation immunoassay platform that you can benefit from, and definitely look forward to sharing more in the coming months. Thank you very much.
Thank you for that presentation, Dr. Bourdon. We want to get right to your questions and input. So here's a reminder as to how audience members can communicate with us. Questions can be submitted via the Q&A button at the lower left. If we are unable to get to your questions due to time constraints, Thermo Fisher Scientific will reach out to you via email. And our first question is, can these assays be multiplex?
That's a very good question. To put it bluntly, no. But let me say this. So whether or not you're performing a classic multiplex immunoassay, for example, trying to measure 30 to 60 analytes in a single well, no. ProQuantum cannot do that, and it wasn't designed to do that. We have an amazing bead-based technology called ProQuantum. It's based on the Luminex backbone, and we should be the customers should be going to that type of immunoassay platform first. Definitely, it's very powerful. Right now, we have a 65-plex that can actually be customized up to 80-plex, and so ProQuantum definitely does not replace that. Now, what it can do, though, is because it only uses a very small amount of sample, and so in the examples I've shown today, you can imagine sort of combining the two different scenarios. So let's say you take 5 microliters, dilute it to a total of 50, but then you do a 2 microliter assay. So without accounting for any slop or excess you might need for pipetting, in theory, you can run 25 replicate wells. So let's say, to be safe, you could run 20 replicate wells with that single 5 microliter of diluted sample. So you can actually perform those assays in parallel. And because we are offering a 384 well format, you can imagine you can fit a lot of wells on 384 well plate. So it's not true multiplexing in the exact same well, but it's important to note that when bead-based multiplex assays are made, like a Luminex platform-based assay, sometimes compromises have to be made on which antibodies to use, because you also have to make sure that there's no cross-reactivity, because you're trying to measure each of the analytes all together in one well. Well, sometimes you may have to pass on certain antibodies that could give greater sensitivity. In the case of ProQuantum, we don't have to worry about that. We're able to settle on the best antibody that not only gives us the great performance in terms of sensitivity, but also meets the strict criteria for spike recovery, parallelism, etc. So definitely keeping the detections separate can sometimes be beneficial. And so there is a way to do parallel measurements.
Does no wash step cause higher background or increased nonspecific binding?
You know, that's a natural question. A lot of people see that cartoon that I show with the two antibodies and the oligos in free solution. And people just naturally assume that if the ligase is present with those two antibodies in free solution, you're just going to naturally ligate those two oligos together even when the antibodies aren't bound to analyte. And the answer is that there's very little of that happening. Of course, the no protein control, that's the amount of signal you get in the absence of protein. So we do account for removing out the background. With any assay platform, of course, there's a minuscule amount of background. But no, the two oligos do not ligate to each other in the absence of protein because, again, it's that stabilization of the antibodies and the analyte. It requires everything to slow down so that the two oligos can be joined together with the DNA ligase.
It looks like we have time for one more question. Does sensitivity improve with overnight incubation?
You know, that's a good question. It depends upon the assay. So if the assay is utilizing two monoclonal antibodies, we find that there's very little improvement in performance by incubating overnight. Now, when I was showing the workflow slide, what I should have said, though, is, yeah, it's very compatible. It's very possible. A user can perform that incubation either at 1 hour room temperature or just naturally place each plate at 4 degrees overnight. It's never going to hurt the assay. And so in the case of mono-mono pairs, we see that the sensitivity improvement is negligible. However, if a particular assay is using a monoclonal and a polyclonal antibody, we've actually seen that, yes, there can be some improvement by performing the assay, the incubation overnight at 4 degrees. Now, just to let everyone know, the performance criteria that we are reporting is always for the 1 hour assay. So in particular cases, yeah, the end user can try incubating at 4 degrees overnight. You may get an improved sensitivity over what we already have, which is very good. But again, it depends upon the assay. And yeah, so I think it's important to note that incubating a little bit longer can never hurt. It's just in particular cases like a mono-mono pair, it doesn't really improve it at all.
Thank you again, Dr. David Bourdon. I would also like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand until May 2018. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay. We encourage you to forward that announcement to your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you next time. Goodbye.